Which of the following procedures would be least likely to prevent bacterial synthesis of the superantigen protein? So first of all, we're not quite sure what this is, but this is some sort of bacterial protein. And here we just got to go through the answer choices and figure out which of these is going to be least likely to prevent synthesis of a bacterial protein. So if we go through these, A says adding tRNA nucleotides that can bind to mRNA and bacterial ribosomes. So that already sounds like the right answer because, that sounds like what you would normally need to do to actually have expression, right? You need tRNA to bring amino acids to mRNA so that the ribosome can integrate them into the growing chain. So I don't see why that would prevent bacterial synthesis of protein. If we look at B, adding a repressor protein that binds to the operator site of the bacterial superantigen gene, well, remember that a lot of bacterial genes are actually organized into operons, which are multiple genes controlled by the same promoter. And these promoters also are associated with basically regulatory sequences called operators where repressor proteins can bind and stop transcription. And so this would indeed reduce expression of the gene, right? Because if you put a repressor protein on the operator site, it will prevent the RNA polymerase from transcribing the genes of the operon. So that sounds wrong, right? Because this would not be the least likely thing to prevent it. It would actually be preventing it. C, adding a specific complementary nucleic acid sequence that can bind to mRNA transcribed from the superantigen gene. Well, again, that sounds like it actually would prevent it, right? Because this sounds like it's describing some sort of like RNAi-like system, where RNAi is basically where you have like microRNAs, okay, or silencing RNAs that are complementary to messenger RNA sequences, and they can anneal to them and basically either silence and prevent the ribosome from binding or actually signaling cleavage of the mRNA, right? So this sounds like a system similar to this. So this would prevent mRNA expression, okay? So that's wrong. And then D, adding a stop codon within the gene. Well, if you add a stop codon in the coding sequence of the gene, you would prematurely stop the ribosome, so the whole protein would not be synthesized. So again, that would prevent normal synthesis again, so that's wrong. So the one that wouldn't prevent normal synthesis would be A.